podcast number 22. And we're going to talk about some eukaryotic organelles, some famous ones, the mitochondria and chloroplasts. And these are, again, found in eukaryotes. So electron micrographs of mitochondria and chloroplasts usually show this sort of thing, where there's a membrane on the outside and then there's membranes on the inside. It's important to tell you guys that this, in order to see things at this kind of resolution, the electron microscope, what scientists do is they slice through it. So you're not seeing a 3D picture. You're seeing a slice through a cell, and this thing is actually much more dynamic in real life and changes shape all the time. If you think about like the blobs in a lava lamp, mitochondria are always changing shape. But if you freeze a cell in one unit at a time and then slice through a blob in a lava lamp, you might get a profile that looks like this or looks like that. Okay, so with that thing to keep in mind, how many mitochondria membranes are there? There's an outer membrane on the mitochondria, and the outer layer has two membranes, okay? And then this inner layer connects to this inner membrane here, extends into the membrane. So there's one layer on the outside, one layer on the inside that folds in like this, okay? So this would fold in like this, and so on. So it's, and then the outer layer here is harder to see, but there's two membranes essentially, and we'll talk more about that when we talk about how mitochondria make energy. And then chloroplasts actually have three membranes. So in their outer membrane, there are two membranes, two outer membranes, and one inner membrane that is all highly folded up. So mitochondria, two membranes, one on the outside, one on the inside. Chloroplast, three membranes, two on the outside, one on the inside, all folded up. Okay. If we look at a single-celled organism that can photosynthesize, is Chlamydomonas, and we slice through that organism, there is a cell wall on the outside. Cell wall is not the same as a cell membrane. It's not made of lipid. It's usually made of carbohydrate, sometimes made of other things. But inside that cell wall is the plasma membrane. Plasma membrane, same as the cell membrane. And then inside of that are mitochondria. Okay, each one of these would have two membranes in yellow. And then there's a nuclear envelope that is two sets of membranes. And then the chloroplasts, there's a profile of chloroplasts here and here. They would have three sets of membranes. So lots and lots of membranes inside a eukaryotic cell. Okay, and there's an outline of all those different kinds of membranes in this single-celled organism that is a version of a plant that can photosynthesize. Okay, in a chloroplast, there is actually DNA that codes for the genes to make the chloroplast. Here is the shape of the DNA, and you notice that it's circular, not linear like we saw with other chromosomes. Okay, and it turns out in mitochondria it's also circular. So where do chloroplasts and mitochondria come from? This is an idea that is not all that old. It's about 40 or 50 years old, so that's not old. If we're talking about the parts of a cell, you would think we would have figured that out a long time ago. But this is an idea that chloroplasts and mitochondria used to be free-living organisms all by themselves. They got taken up by eukaryotic cells and now live as symbiotes inside the eukaryotic cell. So they used to live on their own and now live inside a eukaryotic cell. Okay, so when we are making a tree, we talked about trees a couple podcasts ago. A tree is a hypothesis in that this is a guess about how these organisms are related to each other. And when you're making that guess, sometimes you have to guess in terms of, I think this happened versus that happened. Like, I think this change happened after that change happened in this DNA sequence. When you are doing a complicated tree, a more complex tree like this, you can see what does the tree look like when you make different kinds of guesses. And you change your guesses along the way. And then you have this run on a computer, and you have the computer make lots of guesses and say, what does the tree look like when I make this guess? What does the tree look like when I make that guess? And then it can compare all the trees that it made guesses on all at once. And they could say, okay, of all those different guesses I made in the computer, what percentage of the time do I cluster these two groups together? And in this case, it's 100% of the time. In this case, it's 98% of the time. In this case, it's 88% of the time. The other 12% of the time, I get a different arrangement than this tree. But these things are called bootstrap values, and it's a way to give you how confident are you that these two things are related to each other in the tree. Well, I'm really confident in that because it finds them together all the time. I'm less confident about this split. Maybe this group is not always together, and then maybe this group is not always together, because 12% of the time the guesses lead to different sorts of trees. Okay, so that's a bootstrap value. It's like a confidence value in a particular branch on a tree. Okay, so if we're trying to figure out that if mitochondria and chloroplasts used to be free living, where did they come from and what are they related to? So if you look at the sequence of amino acids that code for a protein that is found in some free living bacteria, like these guys, and also found in chloroplasts and also found in mitochondria, we can use that sequence information to try to figure out what organism that is alive today are mitochondria most closely related to. And you can get a signal, in fact, a strong signal, 100% bootstrap values, that mitochondria are related to this group of bacteria and this group of alpha proteobacteria. And you also get a strong signal that chloroplasts are related to a group called cyanobacteria that are another kind of prokaryote, photosynthetic prokaryote. MRCA stands for most recent common ancestor. So these are the most recent common ancestor of modern mitochondria. And the most recent common ancestor of modern chloroplasts is this cyanobacterium. Okay, so we talked about a couple organelles now, nuclei, mitochondria, and chloroplasts. What lines of evidence support the origins of each organelle? Okay, eukaryotic nuclei have histones in them, and those histones are not found in bacteria, but they are found in archaeans. So it's likely that a nucleus or something like a nucleus was in an archaean before it was in eukaryotes. So the origin of nucleus might be in an archaean prokaryote. The origin of mitochondria in that they have circular DNA just like bacteria. And they don't have histones. I didn't mention that earlier, but they don't have histones just like bacteria. Same is true for chloroplasts. The idea that they have multiple membranes is the idea that sometime in the history of eukaryotes, the eukaryotes swallowed up, one cell swallowed up another cell, and the multiple multiple membranes are left over from one cell swallowing another cell, surrounding itself with its own membrane, and then that lives inside the mitochondrial chloroplast. So that sounds like it's pretty far-fetched, but it's not so far-fetched. There's tons of evidence. Mitochondrial chloroplasts are about the same size as bacteria. They can divide independently of the cell that they're sitting in. So if you work out, you get more mitochondria typically in muscle cells, even though you don't get more muscle cells. Your muscle cells get bigger and have more mitochondria in them. Plants vary the amount of chloroplasts that they make depending on light intensity, and they do that without splitting the cell. There's also some animals out there that can actually do what we think happened in evolutionary history. So there's a sea slug called Aplysia, Aplysia, and it will feed on algae and then extract its chloroplasts without digesting them. And instead of digesting them, they put the chloroplasts on their back, and they're actually green. And this animal, even though it's an animal, can photosynthesize with these stolen chloroplasts from the algae that they eat. So we know that it's possible for one organism to eat another organism without digesting it, and for it to take its organelles and actually use them. That happens in this sea slug called Aplysia. Okay.